organisms that glow in the dark using fluorescent proteins. Now they promise to revolutionize medical science. A cellular observation technology called bioimaging could light the way to new cures for disease. For example, a way to make only cancer cells glow to show exactly where cancer is spreading. Or we may be able to illuminate the way visceral fat triggers strokes and heart attacks. Get ready to explore the frontiers of bioimaging, a technology that is changing the future of medicine. Hello.
Hello.
This is Science Zero. Today's topic is bioimaging.
And speaking of bioimaging, have a look at this. Equoria victoria. In 2008, Osamu Shimomura was awarded the Nobel Prize for his work with this jellyfish.
Yes, with its beautiful green glow. Well, fluorescent proteins, such as the one found in this jellyfish, can be used to make living cells light up. That's what bioimaging is.
In my research, I study the flow of blood throughout the body in relation to diseases of the circulatory system, diseases such as arteriosclerosis. So fluorescence techniques are critical to my experiments. They can help me to see how the flow of red blood cells, for example, is affected by disease. I'm eager to see the images other researchers are capturing using these techniques.
First, take a look at this. Here, the blood vessels and blood plasma have been made to glow in different colors. Red blood cells appear in black and white blood cells in light blue. Green shows platelets.
Incredible video. It's so colorful.
Yes, it is. It's also incredible that we can see real-time imaging of activity inside a living organism.
On today's program, we'll be looking at how bioimaging promises to transform medicine.
We'll begin with how bioimaging could change the treatment of cancer. Let's take a look.
A stomach cancer patient is undergoing surgery. Advanced stomach cancer tends to metastasize into the peritoneum, liver, and other organs, so surgeons need to determine whether it has spread before cutting out the cancer. Biopsies are taken of tissues where metastasis is suspected. The tissues are frozen and thinly sliced. Then the samples are studied under a microscope to determine whether cancer cells are present. This usually takes 15 to 20 minutes.
When we are in the middle of a biopsy, we must suspend surgery until the biopsy results are available. Unfortunately, when we stop surgery in this way, it can be very hard on the patient's body.
Professor Tetsuro Takamatsu is developing a method of spotting where cancer has spread without interrupting the surgery, by making only the cancer cells glow.
When I started my research, I suspected there might be a molecule in normal cells that also has the unique property of accumulating more in cancer cells. That's what I was looking for.
Takamatsu-san homed in on a molecule called PP9 that occurs naturally in human cells. PP9 is known to emit red light when it is illuminated with blue light. Takamatsu-san wondered whether it could be used to diagnose metastasized cancer. In normal cells, PP9 changes into a substance called a heme group. But in cancer cells, the activity of the enzyme that transforms PP9 into a heme group is diminished, so the concentration of PP9 builds up. Before operating, Takamatsu-san gives the patient a compound that turns into PP9 inside the body. He hopes to raise PP9 concentration and make cancer cells glow more strongly. Let's return to the operating room. The lights are turned off. An endoscope is inserted to see if the cancer has spread to tissues around the stomach. This video is from a case in which the cancer had spread. When blue light is shone on the peritoneum, a red area appears. It's less than 1 centimeter across. The cancer has metastasized. It has spread to another spot on the peritoneum.
Seen with the naked eye, it looks white. But that doesn't mean all of the areas that appear white are also cancerous. Only the areas that glow red are cancerous. Areas that look white but don't glow red are not cancer and can be left alone. Until now, we took biopsies of both and waited 20 minutes for the results. Now we can find out instantly. That's a huge advance.
Diagnosis by bioimaging eliminates the need to suspend the surgery and makes it possible to immediately identify the precise location where the cancer has spread.
Without having to take a tissue sample, just using special lighting, they can find out in real time. Amazing.
Yes, because no biopsy is necessary, there is less of a burden on the patient, and the diagnosis is instantaneous on the spot, so the doctors know how to proceed. It seems to be a very useful technique.
There are many such advantages to bioimaging, so let's bring in an expert to tell us more.
Professor Tetsuro Takamatsu of the Kyoto Prefectural University of Medicine conducts research on medical applications of bioimaging. Is this method of detecting cancer becoming standard practice?
Among Japanese, we see a high proportion of stomach and colon cancer cases. So we decided to try using bioimaging with abdominal endoscopy to diagnose cancer. We're the first in Japan. Right now, this method is still at the clinical research stage. We began this stage in 2009 and we have worked with around 30 patients so far.
And how are the results?
Well, until now, biopsies have been taken to determine whether cancer is present in the tissue or not. But with that method, if there is any error in where the biopsy is taken, the cancer will not be detected. With our method, even tiny spots of cancer glow. That means surgeons won't overlook them. It removes the need to make unnecessary excisions. That is much better for the patient.
That's important.
Takamatsu-san's research seeks to apply bioimaging technology to early detection of cancer. Let's have a look at a graph.
Now, when tumors form, they grow rapidly up to about 2 millimeters across. This takes 1 to 4 years. After that, they grow very slowly over a long period of time. This takes between 14 and 21 years.
The growth flattens out.
At present, many tumors are found after they grow to 1 or 2 centimeters. After that size, they eventually grow rapidly and become difficult to treat. Many of the familiar forms of cancer, for example, stomach cancer, colon cancer, and lung cancer, are cancers of the epithelial cells.
I never knew about that period in the middle when the tumor is growing slowly. And obviously you worry about metastasis.
Well, around the transition from this extremely slow growth middle stage to the fast growth final stage is when metastasis begins.
Ah, so if the tumor could be detected at an earlier stage.
Well, yes, if the cancer is removed before it can spread, then the prognosis for the patient will be much better. In other words, if you find a tumor that is still just 2 or 3 millimeters, then the tumor has not had the chance to metastasize. This means that we can completely eliminate the cancer. We hope to catch cancers at an early stage while they are still small.
To identify small tumors, Takamatsu-san focused on a molecule that works inside cells' mitochondria. The molecule is called NAD. It is active when mitochondria are producing energy. If there is insufficient oxygen present, NAD transforms into a different molecule called NADH, then changes back when the oxygen supply resumes. When NADH is illuminated with a certain wavelength of light, it fluoresces faintly. Takamatsu-san set out to exploit this property to find small cancer cells. Here we see cancer cells. Because cancer cells grow rapidly, the growth of blood vessels lags behind, causing chronic oxygen shortages. For this reason, the molecule NADH is present in higher concentrations in cancer cells, so they should fluoresce more. Takamatsu-san experimented extensively on rats with colon cancer.